For those of you who don't know me, I'm Carol Greenwood. I'm with the Department of Nutritional Sciences and have recently stepped in as acting chair. And one of my very first activities is to have the delight of welcoming everyone to the Edna Park. For those of you that are not aware of the history of the Edna Park, this is actually a lectureship that we've had in the department for many years that has been supported by our alumni. It provides us the wonderful opportunity to bring in eminent nutritional sciences. Our graduate students have the opportunity to interact with scientists during the day, and it ends in a celebration in terms of having alumni and current trainees listen to some recent work that's going on. So we are very grateful to the alumni in terms of supporting this activity. The other thing that we take advantage of in this day of celebration is to acknowledge our award winners. And we have a number of awards to hand out. And excuse me, one of the series of awards goes to our Masters of Public Health students. And given that we're in the building for the Masters of Public Health, we thought that it would be quite appropriate that we would start our award ceremony with those students. So, Anne, if I can ask you to come up.
Thank you, Carol. Maybe sure, I'll hand them to you and you can hand them out. Right. How's that? Okay. So the first award is the Barbara McLaren Award, and this is an award that's given to the top-ranked student after first year in our MPH program, and this goes to Megan Charlish. The next award is the Danielle Hawes Graduate Scholarship, and Danielle was a real pioneer in the field of dietetics education. She was my internship director way back when at St. Michael's Hospital, and when she passed away, her family initiated this award. So I'm very thrilled to present this award to Danji Cheng and Muna Osman. I'm not sure And I don't see Muna here. She's not coming? Okay, I'll hang on to it for her then. Thank you. The next award is the Heather Milne-Nielsen Scholarship. Some of you might have known Heather Milne-Nielsen was a former director of this program and a leader in public health nutrition. And this award goes to Megan Charlish and Christy Docking. And the Grace Waymark Morgan Award is shared by 3 students, and this award is given to students who show promise in the field of public health practice. And this goes to Nazima Qureshi, Mari Somerville, and Christina Bienick. Actually, and that's it.
Thank you so much, Anne. At this point, we're going to turn to our undergraduate scholarships. And Fiona, I gather you're going to give me a hand with, or, yeah, handing these out. We have a series of here. So our first undergraduate scholarship is called the Angeline Stevens Bursary, and this was established by the estate of the late Angeline Stevens, and it's given to our third-year students, and it's awarded to students enrolled in our specialist program in nutritional sciences, and this one is based on academic merit, and we have 6 individuals that we would like to honor with this one. So the first is Chu Chul Chow. I'm not sure if, and I'm sorry if I have the names wrong. The next one is Yung Huang Fu. Thank you. Alicia Kazakova. Jarvis Noronha. Congrats. Jordan Spradbrow. Thank you. And Q Wei Richard Yang. And I apologize if I've mispronounced anyone's names there. And given that I teach fourth year, I look forward to seeing you folks next year. The next award is the Anna Howe Reeve Scholarship. And this was established through a donation from Dr. Reeve in memory of his wife. The Professor Emeritus Donald McGregor of the Department of Economics, the grandnephew of the donor, has subsequently contributed, so it's been ongoing for a while. Again, it's for our third-year undergraduate students, and this one is awarded to a student who has demonstrated general proficiency in third-year courses offered through the Department of Nutritional Sciences, and this year's winner is Catherine Brunstein. Congrats. The next is the Annie Laird Prize in Nutritional Sciences, again a very familiar name I think for many of us, and this award was established through the bequest of the late Professor Annie Louisa Laird, and she had been head of the Department of Household Sciences at the University of Toronto from 1906 to 1936, and this goes to a second-year student with a grade A standing in the second-year courses required as part of the specialist program. And this year's winner is Jiang-Ying Liu. Congratulations. We then have the Charles Ganton Webster Prize in Nutritional Sciences, and this one was established through the generous gifts of friends in memory of Charles Ganton Webster. This is for a fourth-year student who, in the best judgment of the department, best fulfills the undergraduate research challenge in nutritional sciences. And if I can have Zilla Samani Azad to receive her award. Thank you. The Clara Benson Scholarship is dedicated to food sciences. It was established by the Household Sciences and Nutritional Sciences alumni from the University of Toronto, so we thank you for that. And this again is for a third-year student who's demonstrated a high level of proficiency in the third-year courses of food chemistry offered by the department. And this year's recipient is Rakita Gupta. Congratulations. We have the Daniel Wilson Scholarship in Science, again established through a bequest of the late Charlie Christie in honor of Sir Daniel Wilson, president of the University College from 1890 to 1892, and then president from 1889 to 1892. And it's awarded to a third-year student, in nutrition and food science. And this year's award is going to Chui Chul Chai Chow. No, it looks like either I've mispronounced it so bad that nobody recognizes their name and she wasn't able to attend. The next is the Dorothy Whiting Scholarship in Nutritional Sciences, and that was established by the estate of the late Dorothy Whiting. And this is for a third or fourth year student who makes significant contributions to others through volunteer service to student organizations or the community at large. And Vivian Chu, if you can come and accept your award for this one, please. Thank you. The Dr. Tom Francis Book Award, and many of us will remember Tom Francis from the department, was established by the department, and that it was on the occasion of Tom's retirement. And this is for a third-year student who has achieved high standing in food chemistry and laboratory course in biochemical techniques, really honoring Tom's dedication to laboratory science. And this year's recipient is Linda Badali. Okay, we'll make sure that that gets passed on. The Edward Blake Scholarship in Nutritional Sciences was established through a gift of the late Honorable Edward Blake, Chancellor of the University from 1876 to 1900. And again, this one is for a second-year student, and it's awarded to a student who has completed the core courses required for the second year of the specialist program in nutritional sciences. And if Julia Hanna can come and accept her award for this. No, I'm going to pop that one over. Next is the Household Science and Nutritional Sciences Alumni Scholarship. And for many of you may not be aware that the Household Sciences faculty used to be right across from the museum in what now is a Club Monaco building. This was established by the Household Science and Nutritional Sciences Alumni Association. And here's for a third-year student in the specialist program in nutritional sciences who's excuse me, demonstrated a high level of proficiency in the nutritional sciences courses required for the third year of the program. And if Kwei Richard Yang, I believe, yep, fabulous, can come and receive this award. And this is the Household Sciences and Nutritional Sciences Alumni Scholarship, again established through our Alumni Association. And this one is for a fourth-year student in the specialist program who's also demonstrated a high level of proficiency in our program. And if Sophia Sagaidak could come and receive her award, please. Thank you. So we now have the Doreen Smith Vandervoort Graduation Scholarship, and this was established by the estate of Mary Doreen Smith Vandervoort, former head of the Department of Food Chemistry in the Faculty of Household Sciences. And this is for a fourth-year student demonstrating a high level of proficiency in fourth-year courses taken as the part of the specialist program. And we have 3 recipients this year: Vivian Chu, Shauna Kim, and Shida Nur Hosseini. We again have another scholarship which was established by Doreen Smith Vandervoort, but this one now is for the third-year students, demonstrating a high level of proficiency in third-year courses offered by the department with emphasis on courses in the area of food chemistry. And this year's recipient is Kwei Richard Yang. So I have to be starting to learn how to say your name properly. The Sarah Kennedy Scholarship was established through a request of the late George Kennedy, and this is for a fourth-year female student standing first in the composite mark of at least 3 fourth-year courses offered by the department. And this year's recipient is Sophia Sagaidak. Sophia, how do I say your last name? You know who I mean. Thanks. We have a prize from the Chemical Institute of Canada, and this is a merit award given to a student with high academic standing in chemistry. And the recipient of this award will attend the SCI CIC Awards dinner. So congratulations to Kwei Richard Yang. So that is the end of our undergraduate award. So let's give a big hand to all of our recipients. We now have two awards that are reserved for our graduate students. The first was set up in recognition and honor of Michael Archer. This is Michael Archer is the third longest chair in the Department of Nutritional Sciences, quite an honor. And that the award was established to recognize a student who demonstrates excellence in research and is based on manuscripts that they have published over the course of their degree program. Christina Wong is the recipient this year, and oh, fabulous. And then finally, last but not least, I'll ask Lillian to join me at the podium. And this is an award that was actually established in Lillian Thompson's honor upon her retirement from the department. And this award was to establish recognition of an outstanding student seminar presentation, really in recognition of Lillian's wonderful achievements for education in the department. And Julie Mason, if you can come and receive the award. So thank you and congratulations to all of our award recipients. With that, I would like to turn the rest of the talk over to Paul Penchertz, who has been instrumental in terms of helping us with the oh, I am sorry, Fiona. Yes, my apologies. Yes, it was. Because the award, of course, this whole event is dependent on the gracious cooperation and help from the Alumni Association. We would like to hear more about the activities that follow that, and from there we'll pass over to Paul. Yeah.
Thanks so much. I wanted to welcome you all here this evening on behalf of the Household Science and Nutritional Sciences Alumni Association. You heard a little bit, but I'd like to give you just a wee bit more history about this lectureship. It was established by alumni in 1974 to honor Professor Edna Park, who was considered an outstanding teacher by her students. Edna Park graduated from the University of Toronto in 1917, and after heading off to Columbia University, where she completed her master's degree, she returned to U of T, where she taught until her retirement in 1962. She remained active as an alumnus and an extremely strong supporter of the university until her death in 1982. I was fortunate enough to meet Professor Park while I was a student at the Faculty of Food Sciences in the early 70s. Professor Park was a consummate volunteer recruiter, and that's how I came to know her. Plus, she attended all of the alumni events that she could during her retirement time. She was an amazing ambassador for the university, and I know that she felt extremely honored by this lectureship. We appreciate the efforts currently of the Department of Nutritional Sciences and the Office of Advancement here at the university for continuing to make this a great event on an annual basis and ensure that her legacy continues. So I'd like to now call on Dr. Paul Penchertz to introduce our speaker this evening.
It gives me very great pleasure to introduce Professor Nancy Butte. And I've known Nancy for a great many years, probably since the 1980s. And she says that she came as a postdoctoral fellow to the Children's Nutrition Research Center, Baylor College of Medicine, in 1980. And not very long after that, I was asked down to be a reviewer of the CNRC, and that's when I first met Nancy. She and I have worked together on the macronutrient DRI. She was a leader in the energy side, and you'll hear more about that, and I played a role in the protein side. Now, one of the things that I have been down to the Children's Nutrition Research Center, which is a very impressive thing, and it has really marvelous equipment to measure energy expenditure. There are whole room calorimeters you can really spend a very comfortable day or night in. And there is equipment to measure body composition in detail. All of those things are necessary to do the kind of work that Nancy is doing. She did, first of all, deal with normals. And the definition in the macronutrient DRI of energy requirement was not how much food energy to eat, but how much energy to expend in order to achieve a normal body composition. And that's a perfect segue into a talk, because as you can see, she's going to be talking about obesity. She's going to be talking about it in Hispanic populations and trying to understand the etiology of obesity. I came in '78 to Toronto, and I started working on obesity then. And I can honestly say I don't think we're any further forward in treating obesity. Maybe we're further forward in understanding obesity, and I hope that we'll hear from Nancy some insights that will lead us to a better understanding of obesity. So Nancy, please educate us.
Well, I'd like to begin by first thanking you all for this invitation to provide this lectureship today. It's really an honor for me, and I've really enjoyed my day here with you. Have really enthusiastic, great students. And I really thought about, I had several questions today, and I do a variety of work, and I hope I brought the right lecture, because I had a lot of questions on our accelerometer data and some other work that we've done. But I'd like to really present you data from an ongoing study. We started over a decade ago at the Children's Nutrition Research Center. It's called the Viva la Familia study, and we've been fortunate to have this funded now. We're on our third cycle. It's up for renewal, so we hope to continue. I got involved in it, we started the study in 2001, actually. And I have learned a lot about genetics, and genetics is a very fast-moving field, which I'm sure all of you can appreciate. I work with a large group of geneticists, bench geneticists, genetic statisticians, and I have really provided the cohort and learned a bit of genetics on the way. So I'd like to share with you where we've been and where we're going. Unfortunately, there's not a bottom line. We don't have a punchline. It's still evolving. And I can tell you right now, we don't know the gene. I don't know if there will be a gene responsible for obesity. It's most likely many genes. But I'd like to tell you on this journey that we've taken for, as I said, over a decade now. So can everybody hear me okay? I don't think we're using this thing. I think we'll all agree that obesity is surely a complex disease. But what I've learned from the geneticists, that they have a very specific definition of complex diseases. And that is that they are not inherited in a strictly Mendelian manner. They are characterized by the risk to relatives of an affected individual that is greater than the incidence of the disease in the population. And they can involve multiple genes and environmental factors and their interactions. So the genetic architecture of most complex diseases has not been determined. And there's really two competing camps as to what might be underlying complex diseases. The first one is the common disease, common variant hypothesis. And this states that genetic risks for common diseases will often be due to alleles with relatively high frequencies. That is, that there will be one or a few predominantly disease alleles at each of the major disease loci. The opposing hypothesis is the common disease rare variant hypothesis. And it states that there is extreme allelic heterogeneity for complex traits, and that multiple rare variants with moderate to high penetrance may be responsible for complex diseases. So based on the common disease, common variant hypothesis, a lot of research was done recently dominated by the HapMap project as well as genome-wide association studies, which I'll refer to as GWAS. And from this effort across, there's been over 300 loci reported for 70 common diseases. However, I'm sure you all are familiar with the sort of disappointment in this area, is that it turns out that they explain very little of the variance in the traits. And this is true in obesity as well. And various explanations have been put forth for why this might be, that the studies so far we've ignored epistasis, epigenetics, maybe incomplete association, and structural variation. Alternatively, rare variants with moderate or large effects might be contributing to this missing heritability. And so these mutations are more, they're rare because they may have a major effect. They may have deleterious effects. If we look at these two sort of broad categories of variants, the common disease variants are usually identified through association studies, case control, these GWAS studies. Their minor allele frequency is usually greater than 5%. Their odds ratio for that disease is fairly moderate, 1.2 to 1.5. They're characterized by low penetrance. And very importantly, they very likely are in linkage disequilibrium with the functional variant. And it's turned out very difficult to figure out what is the functional variant, let alone its mechanism. Alternatively, the rare disease variants are usually described by resequencing of candidate genes. Their minor allele frequency is on the order of 0.1 to 3%. They usually have a greater effect with odd ratios greater than 2, moderate to high penetrance. And the variants, when they are identified, they're likely to be functional. So now if we turn to the complex disease, which we've been preoccupied with, is childhood obesity. It's been known for a long time that obesity and in adults, children, it's been demonstrated that it's highly heritable. We look at heritability with what's called the heritability constant, and that's just the proportion of the total phenotypic variance that is accounted for by an additive effect of the genes. Okay? And you can see here, regardless of what type of study design is used, whether it's nuclear families, adoption studies, or twin studies, there has been a significant heritability. It's quite variable, and that has to do many times with the type of study design, and it's always highest in the monozygotic twins, as you can see here. But we strongly believe that it's a large genetic contribution, somewhere on the order of 40 to 60% is what's usually quoted. We also know that childhood obesity tracks from childhood on to adulthood. Garn noted way back in 1985 that there's about a 26% persistence of early childhood obesity into adulthood. We know that the most important predictors of adult weight are look at the parents and the grandparents and also what the degree of obesity was at puberty. We know that if at 18, if you are obese, here defined as a BMI greater than the 95th percentile, your chances of being an obese adult are extremely high. And also we know this tracking of BMI, somewhat logically, it does increase with age, as you can see here by these correlation coefficients from early infancy on into adolescence. So childhood obesity can be broadly broken into these three categories. The first one is monogenic obesities. They're very severe, and they're also fortunately very rare. Pleiotrophic genetic syndromes. These are rare to moderately common. And polygenic non-Mendelian obesity is quite common today. So I'd like to look at the underpinnings of these three broad categories with you. The first category are the monogenics. And these are a single gene mutation in humans. They're characterized by early childhood onset obesity. They are all characterized by pretty noticeable hyperphagia. And they involve loss of function mutations, many of them involving the melanocortin system and the melanin concentrating hormone system. Earlier today, when we were talking about leptin deficiency. And this is one child, you can see when he was 3 years of age, weighed 42 kilograms. And he was treated with daily injections of leptin. Here it's showing him as 7. This was identified by Dr. Arali and Dr. Faruqi in the UK. And it was first detected in 2 Pakistani cousins. And both children have been treated successfully. We know that leptin affects reproductive function as well, so there was a big question if these children would develop normally, and they did. They went through puberty just fine. And so this has been a huge opening in this area of research. After this was first described, leptin deficiency, people measured it all over the world looking for other obese individuals with leptin deficiency. And the count is something about only 10 people have ever been identified. So it is quite rare. There are mutations in POMC as well as the prohormone convertase PC1 that lead to severe obesity, and also into the melanocortin peptides, specifically MC3R and MC4R both lead to severe obesity. So the last count, there are 11 recognized mutations that cause severe obesity, which are the single gene mutations that are listed here. The one that has given a lot of attention, and we've actually done quite a bit of work in this in our laboratory, are mutations in melanocortin-4 receptor. And it is considered the most common cause of monogenic obesity. In one study conducted by Faruqi in the UK, in about 500 severely obese children, 5.8% actually had mutations in this gene. So it really has been reported in many populations now. It's not a single polymorphism. There's up to 16 polymorphisms that have been described that can cause this. So this is really a very good example of a gene where the monogenic mutation can cause a very severe type of obesity, but we now are seeing that other polymorphisms in the same gene can also increase the susceptibility and to a milder form of the obesity. And again, it's really quite prevalent in the populations. Here's a young boy that actually had a homozygotic mutation in melanocortin-4 receptor. Now, the next category was the Mendelian obesity syndromes, and I'm sure you see them here in your hospital. And early onset obesity is a common feature of these syndromes, but they're also characterized by dysmorphic features, developmental abnormalities, and learning disabilities. Probably the most well-known one of the autosomal dominant is Prader-Willi syndrome, but there's actually many autosomal recessive syndromes as well, where obesity is just one of those characteristics. And then there's the non-Mendelian polygenic obesity. And as I said earlier, this does not follow the typical Mendelian pedigree patterns, but we do believe it does depart susceptibility with variable contribution from the environment. So many of us who are working in this area are really concerned with the polygenic obesity at the epidemic levels that we're seeing amongst our children. And the approach to try to identify these responsible variants has changed over the years. As I said, we started this over 10 years ago, and at that time, sort of the state-of-the-art approach to this was linkage studies. That was followed by candidate gene studies. Then next came along GWAS studies. And most recently, people have been focusing on DNA resequencing. And all of these advances have been really with the technological advances that we now have at our hands to actually sequence DNA. So that brings us to our Viva la Familia study. So we firmly believe that the childhood obesity in the Hispanic population is attributed in part to genetic susceptibility, but certainly these children are growing up in a very obesogenic environment. And so we set out to, this just shows the obesity prevalence of children in the United States. For those of you who aren't familiar with our data, we have the highest rates of obesity amongst our minority children, with the very highest rates in Hispanic boys and actually African American girls. But amongst the Hispanic kids, it's about 25% amongst the boys and about 20% amongst the Hispanic girls. So our current working hypothesis, and this has evolved, is from our first grant, but now our hypothesis is that rare variants of low minor allele frequency less than 1% with a large effect size will contribute significantly to a population-level childhood obesity. And so our study design is a mixed cross-sectional longitudinal design. The cohort was recruited and studied in 2001 to 2006, okay? So each family was ascertained on actually it was an obese, we've changed the terminology, it was an obese proband greater than the 95th percentile BMI and also greater than the 85th percentile for fat mass. Once a family was identified with one obese proband, we invited all the children 4 to 19 years of age to be in the study. So in genetic studies, you gain a lot of power when you have multiple children, of course. So our family size goes anywhere from 3 to 8 children within the family. Our final cohort had about 1,600 individuals, over 319 nuclear Hispanic families, and the children ranged from 4 to 19. We did very in-depth phenotyping of the children and genotyping of the children and the parents. We also brought the children back after 1 year for a dynamic measurement of growth, where we just did anthropometry and body composition measurements once again. This is just a brief overview of our methods. We did standard anthropometry. We did body composition using the DEXA machine. We used 24-hour recalls for dietary intake. We used a 3-day accelerometer for physical activity. The kids spent 24 hours in one of our room calometers that Dr. Penchars mentioned. And we've done very, very extensive profiling of a fasting blood sample. The genetics, as I said earlier, we started out with the genome scan. We went on to resequencing, and we did a GWAS study, and most recently we're doing exome sequencing. And we applied sophisticated genetic statistical approaches to attempt to identify the variants. So just to give you an idea of who our families are, of course we're in Texas, and this is pretty typical. If you look here, 82% of the children were born in Houston, but 71% of the parents were born in Mexico, 11% Central America, 3% in South America. So it's a fairly recent immigrant families. At the time, you have to think back now, this is early 2000s, but still this is a very low-income cohort that we recruited, and the average family size was 5 with a range of 2 to 10. This is just showing the body composition and the BMI of the kids. So for those of you who don't work with BMI, BMI is not a constant like in the adults. You can see here the average BMI of the children was a little bit over 30 in our obese children and close to 20 in the non-obese. But as I said, BMI increases progressively through childhood. These kids are actually very obese. You look at the body fat, it was approaching 40% in our obese, in the obese children. We assessed dietary intake, and these papers they've been all published, and certainly there's a lot more detail, so I'm just going to briefly mention here. They basically didn't adhere to U.S. dietary guidelines for fat, cholesterol, fiber, added sugar, and sodium. They were adequate in most essential nutrients, except possibly for vitamin D and E, calcium, and potassium. And then as far as quality, they were quite low in dietary variety, as well as fruits and vegetables. And this just shows you the macronutrient distribution for both the obese and non-obese children. At the time, this is about 10 years ago, I was really pretty shocked when I saw the percent of calories that were coming from total sugars and added sugars, as you can see here, 30 and 20% respectively. We measured the physical activity, free living, and not too surprisingly, the accelerometer counts were lower in our obese children. We found that nighttime sleep duration was lower in the obese children in the 4 to 8 year age range. Sedentary activity was higher in the obese children, and in some, the physical activities levels were really suboptimal in the obese children, amongst girls, and in adolescents. And this is, again, some summary data looking at the partition of physical activity in sedentary, light, moderate, vigorous activity during the awake period of the day. And you can see that it was the sedentary and the light that were significantly different between our obese and non-obese children. Moderate and vigorous was not different between the groups. And so we saw sort of an overall pretty low, moderate to vigorous activity in all the kids. This is showing we're very fortunate to have 4 room calometers in our center. And so many times we would have the siblings in each have their own calometer, which really helped with compliance and cooperation of the kids staying with us for 24 hours. And then sometimes we had to do 2 days in a row to fit to accommodate all the children. But anyway, it was an ambitious event for us to actually study 1,000 kids in our calometers. But we did over the period of about 5 years. So in the calometer, for those of you who aren't familiar with it, we're measuring energy expenditure indirectly by gas exchange. And typical protocol is 24 hours in the calometer, and it's standardized what activities they do. And the calometer is really a tool to accurately measure the components of 24-hour energy expenditure. So in absolute terms, certainly the obese kids have higher energy expenditure due to their higher body mass. But we also found that their net mechanical energetic efficiency in cycling was actually lower in the obese kids. We do, with the DEXA, we had fat-free mass and fat mass. So once you correct the energy expenditure for their differences in body composition and body size, we did not see differences in energy expenditure between the obese and non-obese. We did not see any differences in carbohydrate and fat utilization between the groups. And this is an area that's pretty controversial. You'll find papers on both sides of the aisle saying that obese individuals have different rates of fat oxidation. There's a couple things you have to do very carefully to do these studies. Number one, obviously diet has to be controlled, but you also have to know what is the energy balance during your study. And that's seldom known because people don't have access to the room calometer. But the largest predictor of RQ, or your oxidation rates, is your energy balance. So we can actually compute that with the calometry data, so that's controlled in the analysis. When you do that, we did not see significant differences between obese and non-obese. And as I said, we've done very extensive biochemistries on the kids, and I'll just show you a few of them today, and I'll eventually be getting to some of our more recent attempts to do phenotyping. But not surprisingly, the obese kids, actually it looks small, but it's statistically significant because we have a large sample size. They actually had higher glucose levels. But what's markedly increased, of course, is insulin, C-peptide, which results into a high degree of insulin resistance in the obese children. The lipid profile, again, is pretty much as described in the literature, high triglycerides. We didn't see we saw differences in cholesterol, HDL, and LDL, but as you can see, it's not dramatically different between these obese and non-obese children. But remember, they're coming from the same families. They are really pretty much eating the same diets. When we look at the metabolic syndrome or the cardiometabolic syndrome, you can see here the various components of blood pressure, central obesity, high triglycerides, low HDL, higher glucose, and also we added on high fasting ALT as an indicator of non-alcohol fatty liver disease in these kids. You can see that they really fall into the cardiometabolic syndrome. And what surprised me when I looked at this data, as I said, the kids go from 4 to 19. We even had kids who would be classified as having the metabolic syndrome as young as 4 and 5 years of age. So that was really what was surprising to me when we actually looked at this data. We've worked, I have a pleasure of working with Dr. Smith, who's a leukocyte biologist in our center. So we've measured many of the pro-inflammatory and anti-inflammatory cytokines in the kids over the years. And you can see CRP was quite different, elevated in the obese kids. But we did not see significant differences in TNF-alpha. And again, leptin and adiponectin follow the expected pattern of being leptin higher and adiponectin lower in the obese children. We've also measured some other chemokines, as you can see here. And again, we're seeing differences with the eotaxin being lower in the obese children, MCP1 and RANTES being higher in the obese children. The most recent thing we've done, looking at the profiling of the kids, is done some metabolomics. And we did this with Metabolome, for those of you who might be familiar with that platform. And we did it on 803 of the children that were enrolled in the Viva study. This is the instrumentation which is used by Metabolome, for those of you who are. And what I really liked about the Metabolome company is that they really have a very, very extensive library in actually identifying the metabolites. As I shopped around, they've had the most extensive library. One of the first things we do, and our interest here is comparing the non-obese against the obese, is this forest plot. And what is plotted here are the 30 top metabolites that basically distinguish the obese from the non-obese children. And I know that's a little bit difficult to see, but we'll go a little bit further here. And when you do this, you can actually predict with the prediction accuracy just to separate out the obese and non-obese children is about 82% based on these metabolites. So the next 3 slides are actually the heat maps. And here, looking at the differences, these are statistically significant differences between the obese and the non-obese children. And I know many of you here in the audience, there's a lot of interest in amino acids and fatty acids in the school here. So if you look at the general pattern here is that we saw a lot of elevated amino acids and their metabolites in the obese children. And just to point out a few, the branch chain amino acids, leucine, isoleucine, valine, were quite significantly higher in the obese children, following very much the pattern that has been described in adults for adults with obesity and type 2 diabetes. But going down again, the acylcarnitines, the shorter chain acylcarnitines, are higher in the obese. We saw elevated alpha-hydroxybutyrate and alpha-ketobutyrate as well. For the carbohydrates, not too many significant differences here. But also note that if you look at what is measured by Metabolome, it's not as extensive. So certainly it's the amino acids and the fatty acids that are extensively measured that are actually detected, and these are fasting plasma samples. But we see an increase in pyruvate and mannose, and actual decrease in glycerate and citrate in the obese children. And in the fatty acids, again, it's very extensive what is detected in the serum, as you can see here. And the general profile here is that we're actually seeing decreased levels of the fatty acids in the obese children, many of their metabolites, whether it's the glycerylphosphocholene or the bisphosphoethanolamine, are actually lower in the obese children. And if you look over here, again, you can see that the ones that are elevated, in particular, again, some of the acylcarnitines are actually increased in the obese children along with carnitine and choline. So what seems to be evolving when we try to synthesize all this data is evidence for increased branch chain amino acid catabolism amongst the obese children. So as I said, we're seeing higher branch chain amino acids as well as some of their catabolites. We're also seeing higher acylcarnitines, but just some of them. Some of them are higher and some of them are lower. And the ones that tend to be higher are the shorter chain acylcarnitines. And it's the medium to the longer chain ones which tend to be elevated. Decreased. So we don't really know what the cause of this, just the increase in the branch chain amino acids is, but it could be increased dietary consumption, or it could be alterations in energy metabolism or mitochondrial function in the obese children. And here is just showing the pathway. Again, I realize it's a very busy slide, so I tried to highlight for you here in red where we're actually seeing the increase in the obese children. So you can see it seems like there's increased availability of substrate. These are normally converted to their alpha-keto acids, cross into the mitochondria. But then we're also seeing an increase in many of the downstream metabolites. But we don't see an increase in activity at the TCA cycle. Okay? So it seems that there may be an oversupply of fatty acid species that may be beyond the capacity of the TCA cycle and is being shunted elsewhere is one possibility. And you can see this here. If we look at the TCA cycle itself, we seem to see an increase in the availability of substrate, increase in glucose, pyruvate, lactate, but we actually see a decrease along the TCA cycle for citrate, malate, for instance. There's an exception here that glutamate is elevated, which may be feeding this the elevation that we see in the alpha-ketoglutarate. So all these various metabolites are really pointing to potentially decreased fatty acid oxidation in the obese children, as evidenced by the higher levels of the long-chain fatty acids, lower levels of the long-chain acylcarnitines, and the medium-chain acylcarnitines, and lower levels of the decarboxylated fatty acids, their intermediates, which are listed here. And we're also seeing reduced levels of the ketone body beta-hydroxybutyrate in the obese children. So these all taken together are really quite suggestive of decreased fatty acid oxidation in the obese children. And again, this again just showing you the pathways and some of the individual metabolites themselves, starting with palmitate as an example of the elevation in the substrate, but then as you can see here, a decrease in some of the downstream products. So again, this has been possibly suggestive mitochondrial dysfunction in the obese kids. We're seeing a 1.3 increase in the alpha-ketobutyrate, an increase in alpha-hydroxybutyrate. And this may be that we're overwhelming the TCA cycle, possibly, and there's a shift actually from the alpha-ketobutyrate to the alpha-hydroxybutyrate. And again, without further research, we're not certain of where it is, but it does seem like there is an excess of the fatty acids species, and it's kind of they are getting partially oxidized and then get returned out to the plasma in the forms of these acylcarnitines. This has actually been shown in adults, both in obesity and with type 2 diabetes, that the hydroxybutyrate is a good marker for insulin resistance, and we're certainly seeing that in our children as well. We're also, within the metabolomic platform, there's several markers of inflammation. And so in addition to the cytokines that I showed you earlier, there's also markers of inflammation, such as increased bradykinin, elevated xanthine, and elevated uric acid, urate in the obese children. So you do get a lot of data from these platforms, and they're not measuring the absolute amounts. These are relative amounts. So we wanted to further try to synthesize this, so we did some principal component analysis on this data. And we, somewhat arbitrarily, but when you get the metabolomic data, a lot of times you'll get missing in a given sample, and that's just that particular metabolite falls below the level of detection. So we used the cutoff, if there was greater than 40% missing, we excluded that metabolite. So we did the PCA analysis on 304 of the metabolites and came up with 20 principal components at loadings greater than 0.5. And that included 161 of the metabolites from the original batch. These explain about 53% of the total variance in the original metabolites. And what we found is that 7 of these PC principal components were significantly associated with our obesity markers, whether we look at BMI or fat mass or fat-free mass for that matter. And then quite interesting, 5 of those were significantly associated with the metabolic risks, the risk factors, whether it was insulin resistance, appetite regulation, triglyceridemia, non-alcohol fatty liver disease, or hyperuric acid in the kids. And so this is just shown here, again, just to show you the PCs that were related with our obesity markers here on the top. There were 7 that are significantly related. And then if we look at these risk factors for the various comorbidities, you can see that 6 of them were, 5 of them were significantly correlated with the comorbidities. Okay, So that's where we are on our phenotyping, characterizing these children. And so I'd like to switch now gears to genetics. And I'll walk you through fairly quickly what was some of our earlier findings and show you where we are today. So as I said, our first strategy was to do a genome-wide scan. And at that time, in the early 2000s, as I said, the state of the art was we typed 384 markers. And this was done in all the parents and in the children. After that, we then went to doing extensive exhaustive resequencing of positional candidate genes that we had actually identified in our linkage studies. And the next approach was the GWAS approach, where we used the Illumina, where we typed 1.1 million SNPs. And now we are doing exome sequencing across the whole genome. So as you can see, we've gone from 384 markers on to exhaustive exome sequencing. So it has evolved quite a bit. Just to give you an idea of what our pedigree looks like, this is very important because the more related individuals you have, the greater chances you have for identifying the variants, and in particularly rare variants. So very typically we have, as I said, 3 children is the average, up to 8 children in a family, but we also have related families. So we accepted into the study cousins, related parents. So here's 4 families that are related, showing the parents, the grandparents, and the children, the generation. So that gives us several relationships, pairs to look at when we do the genetic analysis. The first thing you do before you begin your analysis, you have to make sure that your phenotype is heritable. And so here's the heritability constants for our body weight and body composition. And as you can see, they certainly are heritable. I get this question a lot. People question if really obesity is heritable. And I'll say, well, do you think height is heritable? And people say, yes, of course. And as you can see here in our study, the height heritability constant was about 0.65. And for our BMI, we're at about 0.4. So I think there's really no question, and that certainly there's a large genetic contribution to obesity. Also, if you look at the cardiometabolic risk factors, they also have a genetic contribution. I think it's obviously a very active field is trying to understand the genetics under type 2 diabetes, for instance. But as you look down here, this list, there's heritability for just about everything we measured has some contribution genetically. This is just a summary slide from our linkage studies. And so, again, for the sake of time, I just made this one summary slide to show you that, indeed, we did detect significant linkage across the genome on several chromosomes. For those of you who aren't familiar with this area, to be a significant linkage signal, the load score has to be greater than 3. And so we really had, for one study, quite a few significant hits. And what really distinguishes the approach that we've taken here is that many studies in obesity, all they look at is BMI. And we firmly believe that detection of the variants will be very important to look at what we call endophenotypes that are along the pathway to the obese state. And there's probably many pathways which end up as the obesity, the final state. But we think it's important to look at the endophenotypes leading to obesity as well. And if you look at many of the GWAS studies, they now are up to 50,000 individuals in some of these very large mega analysis that they're doing. But the only common phenotype that they have for obesity is usually BMI. And so I think they have a limited ability to sometimes be picking up the responsible variants. So I'm just going to give you an example of a couple of these linkage signals. This is what it would look like. This was one signal that we had on chromosome 13. And we got the signal with fasting glucose levels. Okay. And so when you look at the genes that are under that peak there, and so the good thing is it kind of localizes where there might be a gene responsible or contributing to your phenotype. The downside of it, usually that region is pretty broad and usually contains many genes. So at the time we're doing the study, it was really very expensive to try to sequence that whole region. So we had to be selective in what genes we want to sequence. So we looked into that peak, and it was what people called a gene desert. which was to our benefit, there were only 27 genes and 14 were hypothetical. And one really popped out, and that was insulin receptor substrate 2. So that was the candidate, so what we would call a positional candidate gene that we chose to sequence. And we did sequence it, and we found 10 rare SNPs associated with our obesity traits. We then did another type of analysis trying to figure out, of those 10, which is the most likely to be the functional variant. And it was this novel SNP right here. And it was detected in 2 unrelated pedigrees, and there were 5 affected children with very severe obesity. Okay, so this is an example of actually a rare variant, which was quite penetrant in these 2 pedigrees. Another signal that we pursued was on chromosome 18. This is for data that we got from the accelerometers on physical activity levels. And you can see this is a pretty messy linkage signal, right? It's covering a very large area. But under this area, there was one gene that we were quite interested in. That was melanocortin-4 receptor. So we pursued that. And we had actually done some previous work. We had a very severely obese child in our study that we, with Dr. Faruqi and Arali, had sent about 10 DNAs, and they analyzed it at that time. And we found a novel functional, a loss of function mutation, this G55V, in melanocortin in this one young boy. And then we subsequently found it in 6 children within that family. It was in 2 of his half-brothers as well as in a cousin, all the same mutation that caused, again, severe obesity within this one extended family. But in order to pursue that linkage signal, we chose at this time to actually sequence the melanocortin gene in 376 of the parents. And then through that effort, we identified 25 novel SNPs, which we then typed in all the children. Okay? And of the 25 SNPs, 7 of them were non-synonymous SNPs, and these 3 were associated with high BMI in the children. So at that point, there really had been quite a few studies on melanocortin-4 receptor published in Europe, Germany, several countries, but it was mainly Caucasian Western populations where they had done sequencing. So many of the SNPs that we picked up had not been described in those populations. We then did the BQTN analysis and identified these SNPs as being the most likely functional variants. And they were related to, you can see these phenotypes of physical activity, total energy expenditure, and actually the appetite hormone ghrelin. Okay. So up to that point, melanocortin-4 receptor is very well known to affect appetite, and that had been described in the humans. The animal model, and there are knockout models of this, have shown an effect both on energy intake and on energy expenditure. So at least in our analysis, we found that it was related to the physical activity levels and the total energy expenditure in the kids. So we think there is the potential for affecting both sides of energy balance in humans as well. So again, as I said earlier, this is a good example of a considerable overlap between that monogenic form of obesity that I showed you earlier and multifactorial forms of in-child obesity. And again, melanocortin-4 receptor is the most commonly encountered in clinical form of obesity. So any of the clinicians in the audience, this is something that should be considered and thought of when you have a severely obese child that you're evaluating. Okay, so our next approach, after we had done the sequencing of the linkage study and some resequencing, the GWAS platform was coming along. And although I think that we all along believed that probably the most effective way to actually find those functional variants is with resequencing, it was really kind of daunting. We had, as you saw, many linkage signals, and really financially, we didn't have the wherewithal to actually go in and sequence the positional candidate genes. So we thought with the GWAS, we might get a better look across the whole genome with now that at that time there was 1.1 million SNPs. What I didn't appreciate when we first did this is that the SNPs that are on these platforms, they're just for mean allele frequencies greater than 5%. So they just don't capture those rare variants. Nevertheless, we proceeded with the GWAS, You have to set a genome-wide significance level based on our pedigree. You can see here it's 1 to the minus 10 to the 7, because we're looking at so many multiple testing. And from our study, we identified 70 SNPs in 34 genes that reached that genome significance level. And again, I always struggle with how best to present this, because it's a lot of data. So I'll just go through this fairly quickly with you. And I've divided our phenotypes into those that were focused on anthropometry and body composition. And these are where we had significant SNPs for the various traits, as you can see here. And throughout our GWAS, these 70 genes that were identified, some of them we just basically replicated what has been reported in the literature for various genes. Others may have identified a gene that is in a plausible pathway leading to obesity. And others you don't have any idea what the protein might be doing. And that's true in this list right here. The first one here, this INADL, really has not been described in the obesity literature, but there is, this is actually from the fruit fly, it has been related to adipogenesis. So it definitely could have some relationship, but it really hasn't been explored further in other models. You go down here to look at the list at the first one here under energy balance, which we found with the sleep duration of the children. This is an interesting gene. It's actually located in the Prader-Willi region. So for those of you not familiar, Prader-Willi is due to a large deletion, and it encompasses this gene. And Prader-Willi have been known to have sleep disturbances. So again, there may be some logical underpinning in that finding. Here are the various SNPs that were related to our metabolic traits, our hormones, our inflammation markers. And again, there's a variety of known and completely unknown associations here. Our largest hit was with that first gene called is the DARK gene, or the Duffy antigen receptor chemokine. And we found that with MCP1. And I'll spend a minute with that one. Again, if you go down, if we look here, there's the melana down here with the glucose levels, MTNR1B. That's a receptor that's known to have an influence on insulin. So again, there's a logical connection here with our glucose levels. APOA5 is a very, very well-known determinant of triglyceride levels. And as I said, just go down the list. This was a very interesting one here on chromosome 9, the thyroid transcript factor 2. This was a block of 23 SNPs that were really quite well known in thyroid cancer world, but it affected our TSH levels, for instance. As I said, our strongest hit was for this gene called DARK, and it is a cysteine-cysteine chemokine. And again, we found it with the monocyte chemotractant protein 1, or MCP1. And it was highly significant, and so this could be a partial link between obesity and systemic inflammation that we see in the children. This is something that Dr. Vorogandi in our group has just recently been focusing on. There hasn't been a lot of attention to the risk of elevated serum uric acid in obese children, and I think it's now getting more attention. But certainly elevated serum uric acid is a high risk for gout, hypertension, cardiovascular, and renal diseases. And we see elevated levels in obese children. As a matter of fact, in our cohort, about 25% would have been classified as having hyperuricemia. And so Dr. Vorogandi went back and did a GWAS on this marker and actually came up with there's 258 SNPs in the solid carrier protein 2 family. Which has been well described in adults, but she replicated it in our data set with children. So as I said, I think our GWAS, it had been successful in that we certainly corroborated several genes that are implicated from other studies. We identified some novel genes that are in plausible biological pathways, as well as novel genes with really unknown function. But we, as others, will have to say that we really explained a very low percentage of the variance in obesity. And so We, as others, have turned our attention to the rare variants as being a plausible cause underlying the genetic susceptibility in the children. And that really brings us to our current effort, where we've turned to exhaustive, complete exome sequencing. And again, for those of you not familiar with this, is that the exome is about 1% of the human genome. And it's about 30 megabases across about 200,000 exons. So with our exome sequencing that we're doing at Baylor, we'll identify all the non-synonymous as well as the synonymous coding variants, as well as indels, insertion deletions, as well as the small non-coding RNAs, the microRNAs. And at Baylor, we're very fortunate to have one of the human genome sequencing centers, and they've developed a very efficient, high-throughput sequencing pipeline. And so our DNA has gone through the system, and it literally is at that last step right now, which is called the project-level VCF files. So we're very anxious to go to the next step and see how this might relate to our phenotyping. This was a summary that we did. We've sequenced 929 children, so almost all of the 1,000 children. So at this point, when we had 810 children done, this is a breakdown of the different variants that we're seeing in the children. And this is pretty typical when you do this type of work. As you can see here, we identified about 156,000 missense mutations. And you can see the high percentage of them that are rare at a very low frequency, less than 1%. Of those, around 26,000 are predicted to be probably damaging, and about 23,000 are missense mutations that are rare and damaging. There's another 3,000 or so nonsense or premature stop codon mutations, and then an additional 9,000 splicing mutations. And this is, again, just showing the same data, broken down whether we're seeing it as a singleton, a doubleton, or a triple or more mutation within the families. But again, you can see there's really are a very large number of probably damaging would be the ones we'll be looking at in particular to see if it relates to the susceptibility to obesity. We also want to look at the distribution, again, of these mutations in the 11 monogenic causes of obesity. Because as I said earlier, there's now more appreciation that besides the monogenic, very severe mutation that causes severe obesity, we're now finding other mutations that have a milder contribution to obesity. So we looked at the 11, as well as the 32 susceptibility genes. I don't think I mentioned that earlier, but in these attempts to do a meta-analysis on all the GWAS studies that have been done, where there really are 50,000 subjects, they now have come up with 32 susceptibility genes for common obesity. So we took a look at those as well. And again, just again showing you here the breakdown of the various mutations in these monogenic causes of obesity, as well as those susceptibility genes. So our next step is really computationally very challenging. And it is very controversial because people are really questioning. So you have these rare variants, and they're only occurring in a few families and a few individuals. Do you have the statistical power to actually arrive at significant associations? And so I work with a group of talented genetic statisticians, and this is the approach that we are planning to do. And I'll have to know in a year or so what we find. But we start out with the association, straightforward association analysis. But the advantage that we have in our study is that we have family data. So we'll be doing a joint linkage association analysis, so it will give us more power. Also, the group that I'm working with, I'm working very closely with the group in San Antonio. I failed to mention that earlier. It's the Texas Biomedical Research Institute. And they have developed software, particularly to work with families with pedigree data. And this technique here is a Bayesian quantitative trait nucleotide analysis, which statistically tries to identify the most likely functional variant. And we've done a lot of that. We'll certainly be looking at gene-centric as well as pathway-centric burden test. There's also thought that individuals may have multiple SNPs, multiple of these susceptibility variants that add to that they'll have a higher burden. And so that's what that's referring to. And certainly there's a lot about informatic characterization of these variants. There's several software tools, the Polyphen is one example, that will give you the likelihood if that's a non-synonymous SNP and a change in that amino acid, what's the likelihood that that will make that protein less effective or dysfunctional. So it's really a combination of all these approaches that we'll be taking to try to identify now. We've got the data in these exomes related to our phenotypic data. So, in summary, I hope I've convinced you that certainly obesity is associated with metabolic derangements. We see glucose intolerance, hypertension, dyslipidemia, non-alcoholic fatty liver disease in these children. We think we're seeing strong signatures of insulin resistance, increased branched-chain amino acid catabolism, decreased fatty acid catabolism, altered energy metabolism, and inflammation in the obese children. We've confirmed the heritability of childhood obesity as well as its comorbidities. And our working hypothesis now that we've seen a large number of rare, probably damaging missense, nonsense splicing variants in this cohort, and that we think this could contribute to that missing heritability I referred to earlier. And it's more likely that these rare variants are functional and will have a consequence on childhood obesity. So I always like to end with this because even though I'm very much immersed in genetics, we all know that families share not only genes, but also diet and culture and many aspects of lifestyle. So certainly I would be the first to argue that it's both genetic and environmental factors that contribute to the development of childhood obesity and its comorbidities. And lastly, I certainly want to acknowledge my co-investigators on this study. The list is quite long. It's been going on for, as I said, over 10 years. So there's a lot of people involved in this, and particularly my co-investigators at the Texas Biomedical Research Institute. And, of course, our funders. So with that, thank you.
So much about genetics. It was amazing. Questions?
That was a great talk. I'm surprised to see that the FTO gene doesn't come up on any here.
Yeah, well, you know, it actually was in that list of 32 susceptibility genes. It was very small print. FTO has not been significant in our cohort. And I've actually even pulled in the 800 kids that I have exome sequencing on. I pulled it up because I do get asked that quite a bit. There were 3 variants, and there's one that is a non-synonymous variant, and it's in one child. So we'll see. If you're familiar with the FTOs, I said many of the GWAS studies came from Europe and the US, and they tend to be predominantly Caucasian populations. There's been some small studies on Hispanics, but really not, it's in adults. It definitely hasn't been shown to be a major player.
I know you are still working on it. At the end of the road, we can see a way to prevent it. Is there a way of using diet? Have you seen any differences in the lifestyle, or we cannot talk about that yet? But I think the final aim would be how to prevent or how to ameliorate this situation, 25% of these. The city rate is quite high.
Yes, it's very high. Certainly, we would like to see prevention. It's the ultimate goal. And there's a lot of preventative measures being taken in the U.S. And so my focus has been on the obese child. because there are 25% of them. And up until recently, so much of the emphasis has been on the prevention. But I think we can't ignore the treatment of the obese child with hopefully to prevent their comorbidities later in life. So both are important. Yes, we would ultimately like to prevent it in all children. But I think we're going to have a lot of societal changes before we see that. I'm involved in a very large CDC study right now, and we actually are doing both preventative measures and treatment measures in a very similar population in low-income families in Houston and Austin. So I certainly recognize the need, but I don't think we're quite there yet. With these kids, I feel that we've studied them a lot, and not just this study, but many other studies which we've done. There certainly is room for improvement in what we're feeding our kids, and certainly we'd like to see all kids have opportunities to more physical activity.
Ask another question. How in relationship or connected with researchers working on addiction? Because we notice that there is a relation, some predisposition among Aboriginal, for example, to alcohol addictions, and that also has been analyzed as a possible view, possibly to genetic factors. Is that, have you chance to talk to any of these researchers connected with them? Because
I think there are people.
In the genetic.
I think that I'm not personally, but certainly there's other people doing research in the areas of the drivers of appetite regulation. And people are now thinking it's beyond just the hypothalamus and looking at the higher cortical areas where the addiction centers are. People are doing functional MRI and looking at presenting people with different types of foods and seeing their reactions. So I think that's certainly a very active area of research. And I think it will turn out to be very important.
Yeah, I mean, it's just because we got an interest in this. One of my students might be trying to look at RDR or RQ, glucose versus fat metabolism. And you're right, there's a hypothesis that kids are mobilizing fat. Yet, I think you said the RQ was not different. But then on the other hand, you said that there's utilization of fatty acids was diminished. So surely that should show up in the RQ.
Yeah, so what basically what I was showing you is that the metabolomic profile and all those indications pointing towards a problem with fat oxidation. But certainly on a global whole body basis, we don't have evidence from the calorimetry that they were having difficulty, any differences in RQ. And we fed them a controlled diet. They were all, it was a 30% fat diet. And under those conditions, no, we did not see a significant difference in RQ.
Just following up on that, that's very interesting. Did I see you mention the free fat oxidation rate? Did you look at sort of the ratio of fatty acid to the protein rate as a sort of measure of oxidation, or just trying to be that oxidation ratio?
Yeah, this data is new to us, so we did the PCA analysis and we're just trying to synthesize it basically. And I haven't done any of the ratios, but I did come across one paper that did a variety of ratios. So that's a good point. We should take a look at that.
I have one quick question then. I mean, you talked about metabolism, which differ in kids. And there's been some suggestion that amino acids and branched chain amino acids could be an early detector of diabetes, secondary diabetes. I guess that's a Harvard group that's done all of it. So the problem is with all this is chicken or the egg, right? Because you're dealing with these kids. And so as a consequence,
Well, you know, again, I haven't, it will be very interesting to actually look at our data and look at age as well. We're seeing gender differences that are pretty strong in a lot of the metabolomic data. But I really haven't focused much on age, so it'd be really interesting to take some of our younger children compared to the older teens as a place to start, and look at the degree of insulin resistance. In this database, we actually had 4 kids that came in to us with undetected diabetes. They already had diabetes, but all the rest don't have diabetes yet. So we could look at some of those other markers that we have and see how it relates to the metabolic markers. But again, the ones that have been pointed out in the adult literature, and it's the Newgart group from the Research Triangle from Duke, the ones that they have found as being markers for insulin resistance and type 2 diabetes, we're already seeing in these kids. I didn't go and look at kind of the very, very young kids, which I think would be interesting.
Sure, because that would be nice to see if there's any correlation with gene expression as well.
You know, I don't. Again, we did this in the early 2000s, and in the calendar, we actually collect 24-hour urine samples because you use that to correct your calorimetry data. And we certainly had the ability, if the kids would cooperate, we could have, and we didn't. One individual I'm working with on the metabolomic data is in our microbiome center at Baylor. And he's been very interested in looking at markers that may be related to the microbiome. So there may be something there, but no, we don't have stool sample.
Yeah, I was interested in the sleep story that kept popping up,
Mm-hmm.
and I was just wondering if you could explain that a bit more. You know, obviously that's the environmental, situational, family bedtime connection would be an interesting story to look at, and I follow that up with family sleep and
Yeah. Actually, I've been involved in a handful of studies now through our work in physical activity monitoring. When we started using accelerometers, we always had the kids wear it 24 hours around the clock. And it was just easier. We weren't really thinking about sleep studies at the time, but we actually had an objective measure of sleep duration. And so in Viva, as I said, we found shorter sleep duration in the obese children. Well, there's been many studies now that have confirmed that with obesity. So shorter sleep duration and also in older children, which we didn't happen to see. So I think there's definitely something there, and people are really now just trying to understand really what's the cause of it. But this is interesting because these are the same families, so the environment theoretically is the same, and we still saw the difference in that younger group with the obese children having shorter sleep duration.
Yeah, I just wanted to hear from you more directly about a population health perspective here and application. It relates to the earlier question, but so maybe it's about sleep duration and those sorts of factors overall in the population, but your genetic work is still pretty specific to small numbers of these children. So do you see, am I right? Or you covered a lot of territory, but often it explains something in one you found it in 1 or 5. Very few of your 1,000-ish.
Yeah, oh yeah, okay. So certainly, yes, these rare variants are being found in selective families, but they're highly penetrant. You can do computations and say, what does this mean on population level? And we'll just see if these various rare variants. So it could be in the same gene. One family could have a variant, you can have different variants in the same gene. But in overall, that one variant will be very penetrant and important to that family. On a population level, the multiple variants in that gene could contribute sizably to the population level. Does that make sense? Yeah.
Any other questions? Otherwise, thank you very much.
You're welcome.
Thank you you so much. I'd like to again take the opportunity to thank our Alumni Association for allowing us to have Nancy here and stimulate thought in us. And we always end up with food for thought, so I'd like to invite everyone to join us in the lobby area here for a bit of a celebration. And thank you again.